Scolari, and I'm a theorist, and today I will present you the work I did on telomeres in yeast. So the chromatin conformation in yeast corresponds to the Rubble configuration all along the cell cycle. So it's a configuration where you have a symmetry axis between the spindle pod body and the nucleolus, and the centromere are the spindle pod body, and the telomeres are located preferentially on the nuclear envelopes, and they form some association between them. And there, at the telomere, is where most of the heterochromatin is found in yeast, which is not a lot. So heterochromatin in yeast is silenced, and the silencing mechanism is this combination of chromatin-binding proteins and enzyme. That is the first well-described reader-writer modules. And makes these foci that you can see by fluorescent microscopy that are mediated by C3, which is molecules which participate into that, and they are functionally equivalent or homologous to HV1-associated chromocenter in human, for instance, if you want to make a relation between them. And the goal, the long-term goal, is to understand why those foci, to understand what is the structure, the physical structure. And not only their presence, but also their location, because they are located, like I explained to you, at the nuclear envelope. And we want to use a modeling approach that complements experiments by collaborators. So let me take a step back and introduce you a very interesting observation of my collaborators. And this observation is that while the chromatin conformation of telomeres is this kind of multi-cluster level in a wild type, if you overexpress CO3, which is a molecule which participates into this, you get one single cluster located preferentially at the center. And then, since I was working in IC laboratory, we wondered, can we see the effect of this clustering, increased clustering in contact map? But this was challenging, because telomeres and subtelomeres are rich in repeated sequences. As you know, those are hard to map, so it was difficult to see this effect by IC. So that's the first part of my talk. I will explain you how, by doing IC analysis, and I found a way to see the effect by IC of this hypertelomeric formation, hyperclustering formation. So I think most of you already know what is IC. Is a technique that is able to map links between DNA and DNA using cross-link, digestion, ligation, and sequencing. And we identified with Roman Cosul and Giuliano Sigonacci some very important statistical properties of IC contact map that I will explain to you now. So first of all, the role of digestion and pull down by biotin have the effect that if you look at the read coverage of an IC contact map, for instance, for a cutter which is not so frequent, such as HIND3, you will see that it's mostly localized at the cut site, so it's not uniform. And this is very important because it gives the most important bias of IC, that some regions are not mappable at all, like the region between different cut sites. So in the lab, we developed a synthetic sequence that had engineerized cut site, such as they were all equally distant, more or less, in the limit of keeping the genome functional. And this could improve the resolution of IC. Particularly, when you do with IC normally is that you bin the data into equally sized bin, like 5 KB, for instance, and then, or 2.4, and then you look at contact map, and you see that in the wild type, where you had this bias, and in the synthetic, there were much less white stripe. So the white stripe are what impeded us to see, for instance, the contact between telomeres. And we searched for a single parameter that could quantify this effect, like this improvement of our synthetic chromatin. And we figured out that if we take a diagonal, like 408,000 base pair from the main diagonal, and we look at the statistic of contacts, this is the cumulative distribution, so it's different in the wild type. Blue is much broader than in the synthetic region. And we can have a single parameter, which is the coefficients of variation, which is calculating the spread of this distribution over their mean, like the mean number of contact. And this was a constant, independent from a genome distance. So we could use this to quantify the improvement of our sequence. Particularly, if you are more mappable, you have lower coefficient of variation. And then I wonder, can we learn something more universal than this, like looks striking, that is always the same? And in fact, we went to the probability of contact, the probability distribution in function of genomic distance that everybody used to model IC data. But this time we didn't use just the mean, but we also looked at the variability. And we noticed that if you take the variability and you divide the distribution, the full distribution, and you divide by the mean, they all fall on the same identical distribution. And this was quite striking. Like, we wonder why nobody ever looked at this, and maybe also that's a suggestion for modeler. Maybe we should also model this when we do models. But and then we wonder, okay, this is true for this new synthetic strain. Is this true for all IC data set like that we had in the lab for yeast? And we noticed that more or less when it's a wild type, it falls on one line, where here is the linear relationship, and when it's synthetic, it falls on another function. And this is a proof that IC follow the Taylor law, which is an ecological law of nature, but it's just the biases. So when you have a higher coverage, you follow this linearity, so the CV is constant. When you have lower coverages, like matrices which are less covered, you have a different law, which related the mean to the standard deviation. So let's try to understand why there is this transition. Like when you are at high coverage, you are negative binomial. So why negative binomial? In fact, it's just one probability distribution, which is integer, range, positive, and have two parameters, which is what we are trying to model here. Is the most simple one and the most logical, maybe. And then if you have the CV, which I showed that is a constant here, and the mean, which is a function of the coverage, we can relate these two to the P parameter of the negative binomial and R parameter of the negative binomial. So apart of that, there is a condition. Probability should be greater than 0 and lower than 1. So negative binomial works when the mean is greater than 1 over CV squared, and that's this point. So how come that is not always negative binomial? Like, of course, there is another step of IC, that is amplification and sequencing. This will end up in random selection of the read that we align. So lower coverage, this condition should be broken eventually. And that's where we get to the second regime, which is the Poisson distribution. So in a way, the sampling noise, like this Poisson distribution, is what is limiting extracting biological information from IC contact map. And this is a factor defining the IC resolution of contact map. With a little issue. An issue is that the range of contacts in a contact map span over 5 orders of magnitude. So you will eventually go in this regime, for sure. And when you go in this regime, you have to rebin. That's why when you zoom out of these tools that now we have available online, they automatically rebin your matrix because you have to bring things away from this Poisson case. But this is limited, because it's setting the resolution all the same, all in your contact map, while this is not necessarily the best strategy. In particular, you have a lot of information close to the diagonal, and you have very few information far from the diagonal. So when you bin, of course you reduce, let's say that we do row ratio, like ratio between two matrices, two conditions. When you rebin, you get more information far, but you lose all the information close to the diagonal. So we wonder, can we make an algorithm that is able to smear the noise only where it is needed? So that's why we came out with Serpentine, which is an algorithm which is published online, that is making some kind of iterative binning of the matrix, which is local and depends on the local coverage. It's a random algorithm, and it's very important that it's using random numbers to do that, because it's improve the quality of the results. And the result is that before serpentine we have this matrix, after the serpentine we have these two other matrices which are more binned here than there. When you do the log ratio, you can get, you can still retrieve all the variability at small distances, while you get also information at large distance that you couldn't have by doing normal binning in a single matrix. Okay. Now let's go back to our original problem. So we had this formation of hypercluster, and they were not mappable. Now we apply the algorithm we just showed, with the two conditions being the delta CO3, this molecule which makes the glue, and the overexpression of CO3 when you have this hypercluster. And we look at the differences, and we see that there is increased contact straight at the telomeres in an interchromosomal interaction, and their extension corresponded to the extension of the chip profile for CO3. Further, we can accumulate this result by setting the boundaries, and we see that there is a compartment in yeast that was previously not observed in IC, and this compartment is only at telomeres, has this shape. And further, we identified some region inside the chromosome that we thought they wouldn't belong to this compartment, but in fact they do. So there are new set of genes that we proposed that are associated to telomeres, and before they were not observed by microscopy. So now I will present you the ongoing modeling work on these results. And another result, which is kind of related, and it's the formation of telomere fossils in the center during quiescence. Because another condition where you have the hypercluster, as has been shown by my collaborators, is when you pass through fermentation, respiration, and stationary phase. After several days, the clusters are not anymore at the surface, and there are not anymore many, but there is one in the center of the nucleus. And this happens, like I said, over several days of carbon source exhaustion, and results in chromatin, which is much more compact, with the cluster in the center. And this is palm of histone, H2B, which show the chromatin concentration by imaging. So the exercise, the objective is to model three conditions: the wild type, where you have a multiple cluster; the CO3++, when you have overexpression of CO3 and you have one single cluster which forms this one compartment; and the stationary phase, where you have also this single cluster, but in a very different condition. And make two measurements. First one is the radial localization, typically from wild type to hypercluster. You change the distribution, you go from this nuclear envelope to the surface, and the same for the stationary phase. And the number of clusters, which get reduced dramatically in CO3++ and stationary phase. And you know, like at the level of principle, like at the level of mechanism, we were interested in the fact that you have many clusters in wild type, and these might be due to the interaction between attraction of these CO3 molecules and repulsion of everything which is around it. And I made papers about that, and I think that the chromosomal arm can act as a surfactant, and that might be the origin of microphase separation as is observed for telomere, but also for other kind of elements. And further, now there is the surface, the interaction with the nuclear envelope, which we didn't have in my previous work, which gives more interest from the biophysical model point of view, in the sense that this kind of condition, like microphase separation and interaction with membrane, has not yet been studied in physics of soft matter literature. So we performed simulation. We used the Zimmer model, which is a well-validated model for yeast. nucleus is 5,000 bits. It has connectivity, steric interaction. It reproduces the Rouse dynamics and the statics. 16 chromosomes anchored to the spindle pole, 1 nucleolus, 32 telomeres, 1 bit each, and telomere nuclear envelope interaction are modeled. What we add is that 3 times the bits, because otherwise there is not enough bits to model telomeres. So 3 bits each for each telomeres. And we add telomere interaction, which were not considered in this work of Zimmer. And the result is that so far we propose that the normal conditions are close to critical. So that's in fact the only way to reproduce some other modeling results by molecular dynamics, like we are doing molecular dynamics that didn't, is that telomere should associate and dissociate from the nuclear envelope and should cluster dynamically in time. And you know, like if you go to a very strong interaction, telomere association will be stable. If you go to a very low interaction, there will not be any telomere association. So to have this kind of dynamical association, you need a critical condition. And these might look very, very, very narrow from a modeling point of view. So we have to balance all single element just to be there, which is a bit hard. But from the biological point of view, might make sense in the sense that if the cell want to change the amount of clustering easily, or want to change the association with the nuclear envelope easily, you need to be at that point. You cannot be anywhere else. Second, we are able to reproduce collapse to a single cluster upon starvation by keeping the same telomere-telomere interaction. So we don't change the energy of association. That's not why we get a bigger cluster. But we remove the repulsion between chromosomal arm, which is the same mechanism which I was explaining you, might cause microphase separation. So critical condition with chromosomal arm and no chromosomal arm interaction, you get single, very dense phases. So chromosomal arm interaction are important. And of course, we eliminated the attraction between the telomeres and the nuclear envelope because we know that it's not there during starvation, and that's why we get most of our result in zone 3, or in the center of the nucleus. Here is associated to the spindle ball, in fact, body. And finally, we went to the strong regime. So we say, okay, now we make a very strong telomere-telomere interaction, 10 times what we have in normal condition, so 10 times the critical condition. Still, using a Zimmer model, we cannot get one single cluster in the center of the nucleus. In fact, the results we get are very compatible with normal condition, or heavily depends on the initial condition. So if we change the initial condition, they might do this. But then we don't know the initial condition, so what is the point of proposing them without knowing that? We can try different initial condition and tell, yeah. But in fact, as you see, cells are dividing. So I think that the cell cycle is very important for the make of this single cluster. And so we need more experiment. We need to get to know this initial condition or more data analysis. By the way, why they're not making a single telomere cluster in the center? Well, you look at this. This is what I made by IC contact map. There is this kind of contact between telomeres, like outside the compartment, but close to the compartment. And this is a tension. This is a result of tension due to the clustering and the steric effect. And this is in nature very similar to what you get for centromere. Also, centromere have this kind of tension. And centromere make this single fossa inside the cell, but it's always there. Yeast have always the rubble conformation, so there is no issue about the dynamics of formation. It's just initial condition. So with this, I thank you for your patience, for listening to me, and the organizer for inviting me to give this talk, and all the collaborators which contributed to this work or to other work. And you're welcome. Any question?
Vittore, very, very interesting story. I wonder about two things. One, you say you need to you need to switch off repulsion between the arms. Yes. Is this necessary? So you wouldn't get a single cluster if you just switch off attraction to the membrane?
No. In fact, attraction to the membrane drive the cluster in critical condition toward the envelope. But if we just remove that, then we go toward the center, it will still be critical because you have the repulsion between chromosome arm. If it is so necessary to make this switch off, that even in strong telomere-telomere interaction, we don't get a single cluster.
Yes.
And this is biologically sounding, because this repulsion is mediated by electrostatic, and there is a massive reduction of acetylation, transcription and acetylation during stationary phase that could explain this reduction. In fact, it's observed in the super resolution microscopy.
Yeah, that's the surprise, because in principle, if telomere-telomere interactions are very strong, telomeres should start clumping together.
That's what is not happening.
That's what I'm surprised. Why is it not happening? Even on the membrane, they should start clumping, right?
Yes. So, take into consideration that this is the Zimmer model. So you have a stronger repulsion. We are not in collapsed configuration. It's yeast. So the exponent is 1.5, it's very open, chromatin. And consider that there have been several papers in the past and also some more recent that show that when you have a heteropolymer with repelling and attracting elements, the brush effect can cause microphase separation. So it's the interaction between repulsive brushes. Oops. This repulsive doesn't work.
Yeah, no, I understand.
Yes. So it's really making a potential that will not allow things to get together unless you start in that condition.
Okay. Can this be a peculiarity of the Zimmer model, that monomers are very, very big? Sort of?
Yeah, okay, so you can. If you don't have this, it will be very hard to get a microphase separation when it is necessary and when this is happening. And this is the most validated model in yeast, the Zimmer model. And it explains not only the telomeres, but which was not looked at all by Zimmer, but the centromere. The centromere, this kind of pattern which I showed the last slide, is explained by the Zimmer model, by the brush effect.
But I'm just generally surprised that if you have very strong attraction between telomeres, they wouldn't clump together, but you're saying excluded volume repulsion between the chains is so strong that it wouldn't allow this to happen.
Yes, it's so strong that you have a clear entropic effect in the alpha factor synchronization and in metaphase. Like you see the tension in IC contact map, so I think it's quite important.
I just want to continue a little bit on that. I think it's actually very, very interesting. I have two questions, I think. First of all, how robust is the result base? You do something like, for example, minor change on the Zimmer model. Do you still see the same phenomena? And the second is, why do you say this is critical? It's just not sort of a tension between two forces. How do you actually claim that you have this very critical thing? It's not more, it's smoother sort of transition.
Okay, let me start from the first question. So the first question, it's if it's robust. So we still didn't make a huge modification, but we are in the process of doing that because we want to test this question. So it will happen. Like we are going to see how much you have to modify in order to break this phenomenology. I agree. It's a good question. The second question is about the broad, how broad is the transition. So here you have 32 telomeres, which are kind of small, so it's pretty thin. And so it's more like first order kind. So it's not very broad. That's why. And you know, like association and dissociation rates have been estimated in 30 seconds and 8 minutes in wild type. So we have this short time scale at which things come and go together. So excluding active processes that might be doing this, we have to resort on being at the critical point.
And just to conclude, since you brought the subject, how do you know that this is sort of just a kind of thermodynamic process? There's no real active process that may be governing this transition?
We don't know.
Hi, Vittore. Thank you for this presentation. I'm wondering how, if you delete CR3, you're not proliferating anymore?
If you delete CR3,
Yeah,
you don't have clustering, but you have association to the.
Yeah.
It is the less clustered condition in IC contact.
Yeah, and the cells, the yeast, is still proliferating?
Yes,
Yeah,
it is proliferating, yes.
So, okay, so I can ask my question. So you're going through mitosis.
Yes.
And in mitosis, so you condensate the chromosomes, what's happening there? You separate, you clearly condensate and segregate the chromosomes.
It's very complicated, yes. Yes, there's clearly an active process it's driving. So the spindle pod body gets connected to the microtubule, which will push the daughter cell and the mother cell on their respective nuclei, and then there will be segregation. So at that point, the exact location of telomeres, we don't know.
Okay. That's why you're saying you have no initial situation, clear initial situation where to start with.
That's an interesting problem. That's why I raised this interesting problem.
Good.
In fact,
Okay.
yeah.
Okay. Thank you.
Okay.